So dear students, very good morning. I hope I am audible to you. Can you answer me?
Yes, sir. Good morning.
Yeah, very good morning and welcome back for today's session. This will be the fourth part of fluorescent microscopy. So related to this will be the concluding lecture, and after that we'll switch over for the some another new content over there. So dear students, as you know, fluorescence microscopy, this is one of the important advanced technique as compared to the conventional compound light microscopic technique. This microscope basically uses much higher intensity light sources which excites a fluorescence species or fluorophore you can call in a sample of that particular object of our interest for microscopic investigation. So this fluorescent species or fluorophore in turn emits a lower energy light of a longer or higher wavelength that produces a magnified image instead of a original light source. So the enhanced image, it is as compared to the light microscopic image, is totally different. So this was a part of our discussion with reference to its principle, its construction. We had discussed a lot of things in previous few lectures. Now, as you are quite aware about the fluorescence microscopy, let us start with a discussion on applications of fluorescence microscopy. So this is what you have seen in last few lectures, that fluorescence microscope, how it works over there, what how it is get constructed and how it functions with reference to light sources. So as far as concerned with the refinement of epifluorescent microscopic technique and the advancement of more powerful focused light sources such as laser beams or laser light, that had led more technically advanced scope such as the confocal laser scanning microscope, sometime we can call it as a CLSM. In fact, there is a total internal reflection fluorescent microscope has been get invented, that is called as a TIRF. So these are the uh, actual gift from fluorescent microscope itself. So as far as concerned with CLSM, we what we call here CLSM, it stands for confocal laser scanning microscope. These are invaluable tools for producing high resolution 3D images of subsurfaces, more often in specimens such as the microorganisms. Now, these CLSMs has a great advantage is that they are able to produce sharp image. Okay, they are able to produce sharp images of thick samples at a various depth level by taking images point by point. So we can just go on scanning up at different depth level and try to get reconstruct all this depth level in the form of a computer aided image rather than the viewing a whole image through a eyepiece, through an eyepiece. So this is the advantage of CLSM. It is possible, and due to the epifluorescence microscopy, in fact, this is becoming little bit more is that significant. Now, if you look for these microscopes, especially fluorescence microscopes, they are often used for use that imaging then structural imaging the structural components of small specimens, such as you can call it as a uh, microscopic object in the form of a live state itself. So dear students, this part what you are looking on the screen, it is already get discussed, but main important part is that now all together in a summary form we will see that these particular structures like as the microorganisms or very smallest object, okay, they may be get visualized. In fact, for conducting the viability studies on cell populations, they are more often whether in active phase of their metabolic reactions or not, or sometime it may be whether viable or dead. So to check for that, this microscopy is more often used. In fact, imaging the genetic material with a cell, especially consisting of DNA or RNA, that is also becoming possible due to this fluorescence microscopy. And another important part is that viewing the specific cell within a large population with a technique such as a FIS, that is also possible nowadays. So my dear students, this part already were discussed during the previous lecture, and now let us go in exploring a little bit details of this particular microscopic technique. So as you know, fluorescence microscopy, it is used to analyze the biological structures in a sample. So, fluorescence microscopy is a technique which is used to analyze biological structures in a sample using a white lamp and other organic or inorganic fluorophores. That is, fluorophores are nothing but you can call it as a substances uh, emitting the fluorescence. And of course, uh, such fluorophores or dyes, they excites a photoemissive use that reactions, and which is more often observed using the optical band passes. I had told you about band passes in last lecture. That band pass filters, and in fact dichromatic mirror is also get used for this purpose. So various biochemical industries, they rely on such type of fluorescent microscopy tool, and it is meant for the performance of molecular imaging to support different types of medical diagnosis, or sometimes in research also. So to carry out research into a cellular structures and at nano, especially at a nanomolecular activity level, means internal metabolic pathway, they can be get studied by means of such type of a fluorescence microscopy. So fluorescence microscopy plays significant role to analyze the biological structural samples. The another important aspect of fluorescence microscopy is it can be get used for cell labeling. So fluorescent microscopy is more often used for cell labeling. Now what is exactly cell labeling? So this is also a kind of a tracer pathway over there, in which it only important or desired or desired cell organelle or desired cell protein structures that may be get targeted and recognized properly. So labeling of cell structure with a fluorophore, or you simply call them as a dye, such a processes are, especially fluorescent dye, they are drastically increase the observable property of biological or it may be the chemical structures due to this particular nature of a fluorescence itself. So this use of a use that a fluorescent substance or fluorophore is nowadays drastically used in such a type of experiments or in such type of investigations. So these can be get modified, or sometimes it may be get conjugated to exhibit a specific targetability. So this specific targetability is meant for use in different medical applications. So my friends, the fluorophores with high specificity are routinely used in medical diagnostics. So you have to remember this part. So I repeat, fluorophores with high specificity are routinely used in medical diagnostics. With increasing research findings, further applications for fluorescent cell labeling has enhanced also. I'll tell you example. Researchers have successfully used a fluorescence microscopy to track and analyze is that a viral DNA in a cell at a local level, and of course further this is bridged, or it is get connected that to cellular antiviral defenses also. So tracking and analyzing the viral DNA which is present there in a cell, and this particular observation to get connect to a cellular antiviral defense research or investigation that is possible nowadays due to this mechanism of fluorescence microscopy, where we can do cell labeling. So this has very good potential, is that ability to carry out different types of experiments or researches in at cellular level. So dear students, this has potentially tremendous implications for future viral analysis and categorization also. So viral research is more often based on immunofluorescence microscopy. Generally it is nothing but basically fluorescence microscopy by labeling the cellular components, or cell labeling in short you can call. Another part is that what I want to now suggest to you is that this fluorescence microscopy has great advantage in protein characterization also. So fluorescence microscopy is used for protein characterization also. So as you know, the intracellular dynamic protein interactions, they are crucial for a range of critical biological processes, and they are acting as catalyst. More often proteins, they are acting as a catalyst for some, say for example certain metabolic reactions, or it may be responding, that particular protein, it will be responding to stimuli also. So as I say that the intracellular dynamics, it is little bit complicated reaction, either in the form of a enzyme or catalyst it work, or it may be as a stimulating substance or stimulating respond in fact. So in different way these proteins are behaving, that indicates intracellular dynamic of protein protein interaction. It is little bit very difficult. So switching over to a new protein function and switch off to the earlier protein function, that may be process happening within nanoseconds, or you can say that a picoseconds time duration in fact. And to trace out such type of events or cellular metabolism, it is really great job or difficult task. So the protein interactions are typically observable, you have to always remember. So protein interactions are typically observable by using a dedicated instruments only. So those instruments which are capable for analyzing the samples at the nanometer scale or piconanometer scales, so that smallest time duration of event has to be get captured. So such a instruments if you are using at that time only we can do it. However, the transient interactions of proteins that display more often weaker signals. So when we are trying to use such type of instruments which are capable for analyzing the samples at nanometer scale, in fact the transient interactions of proteins displayed there a very weak signal, and which are typically very transparent. And even to it happens in case of a fluorescent microscopic instrumentations also. But uh, as compared to some other sophisticated devices, fluorescent microscopy, it is showing quite better use that detection and gives the best result. So the implication so the implementations of this fluorescent microscopic fluorescent protein biosensors is nowadays considered as a noble approach, because these fluorescent protein biosensors, they are used to observe such type of weaker protein interactions, which more often found at a broader application level in various fields of research. Just now I told you why there is weak proteins, why there is a weaker signals may be get produced, and if you are using such type of a good quality of biosensors which are capable to detect the fluorescent protein interactions by means of detecting that particular signal, which is very weak, but even then it can be get detected due to such a newly discovered use that biosensors. So researchers have utilized the fluorescent proteins to observe intracellular reactions in the hope of gaining insight into various transmission reactions basically. So, by analyzing the specific wavelength emitted by the excited cell during a protein synthesis, this is definitely possible to carry out understandings on protein characterization. So, my friends, such type of studies they have a tremendous potential to provide, of course, insight into how irregular translations in a cell can be result in a developmental problems and disease transmissions. So as you are quite aware, there are variety of genetic disorders are there, and which are basically related with your faulty translation mechanism. And of course, a faulty translation mechanism of a protein synthesis, it is a result of actually at molecular level of a DNA that is mutated over there, and through that such a type of a cellular protein expression, if it occurs, and it causes certain developmental related issues over there, if it is in early embryonic phase or it may be in a mature adult. In fact, various kinds of diseases may be takes place, but if this mechanism at a genetic level may be get understood, the disease transmission to check or observe it may be becomes very easy. But to carry out such type of a test, in fact, in as a part of a medical diagnosis, protein characterization is really very important. If a sample of a patient having such type of a developmental disorders, or if patient is showing certain disease symptoms over there, so protein analysis at a specific target level, if it is carried out by using a fluorescent microscopy, and if you notice there is a some abnormal protein has been get synthesized, that would be a diagnostic tool or diagnostic test you can call for such type of a protein use that a patients over there. So but they also require more exceptionally a very highly sensitive instrumentations, and of course highly tunable chromatic filters to carry out very accurate assessment data over there. So that is only one limitation, but even then we can carry out such type of protein characterization in very excellent fashion. uh, and if you remember, my friends, during previous lecture we came across with an aspect on the fluorescent microscopy that is very extensively being get used for dental research, in fact. So in dental investigations, especially related to the hard tissue, it is definitely possible to carry out detailed insights related with such a tissue. And this is actually one of the latest use that a field of a invention by using the fluorescent microscopy. So as you can see on the screen, very recent article, that means in last 2 decades such type of experiments were carried out comparatively to this new area of a is that a research and application, in fact. So there are many studies have been showed to we people that human teeth, it shows autofluorescence. And of course there are several applications of fluorescence microscopy have been get reported, but although the chemical nature of a autofluorescence, it remains unknown, though the fluorescent microscopy was used, but the autofluorescence nature was remaining unknown in case of a dentistry, this particular field of a tooth science, you can simply call. So due to this uncertain nature or unexplored chemical nature of autofluorescence, recently the fluorescent microscopy was tried for the investigations on a dental hard tissue. So this group of a scientist, that is Matsumoto and his co-worker, they carried out such type of a medical explorations, and this particular research article was get published in one of the renowned yes, that a journal, that is Frontiers of Medical Biological Engineering. in year 2001. So this is one of the important advantage of a is that a technique or instrumentation known as a fluorescence microscopy. So my dear friends, we were talking about fluorescence microscopy, and this is what we had discussed so far. So I hope that you understood this entire mechanism, and if you still have certain doubts or difficulty, you can ask it to me also. So this is related with your the very first sub chapter under instrumentation and technique from unit number 4. I will take certain queries.